Welcome learners to Understanding Biomolecules webinar. This is presented and brought to you by Master of Arts in Biology Education students of Bicol University under Cell and Molecular Biology class of Professor Clarence Faith Bueno. To start the program, let's feel the presence of the Lord for the invocation to be followed by the Philippine National Anthem.
Dear God, thank you for the opportunity to meet together. Please help us to come together to make this institution reflect your kingdom. Breathe life into our ideas and decisions. Help us build a team that has love and respect at its heart. Give us the strength to continue working for your kingdom in this time of pandemic. Lord, come give us the inspiration to be the best we can be. May we be a shining example of your goodness and truth within, wherever we are. Inspire our thoughts, discussions, and ideas, and continue to remind us that all that we do here today, all that we accomplish, is for the pursuit of truth for the greater glory of you. We ask these things in your name. Amen.
Mga kababayan, ang pambansang awit ng Pilipinas.
Kayang magiliw, helas ng sinakanan, alam ng puso, sa titik mo'y buhay. Lupang hinirang, tuyag ka ng magiting, sa manlulupin, di ka pasisigil, sa nagatang tutok sa simoy at sa langit mong pagaw, ay hinatang tulad, awit sa pagkayang minamahal, ang isang ng matawad mo'y tagumpay na nag-iindig, ang bituin at araw niya kailan pang ay di magtitingin. Luman ang araw ng walhagit pagsinta, buhay langit sa piling mo. Ang inigaya na pagkay mga amin, ang mamatay ng dahil sa'yo.
We hope that you are doing well. This time of pandemic has paralyzed almost everything. But being paralyzed or being crippled doesn't mean we stop learning. Hence, as educators, we always find ways to address current lapses. To address and discuss topics that are not covered by modular mode of learning, we present to you this webinar, turned lecture in Biomolecules. Now, this lecture covers 3 parts to be discussed by 3 MaBioEd students. The first part will cover topics in Introduction to Macromolecules and Carbohydrates, to be followed by lipids and proteins, and the third part will be all about nucleic acids. Generally, this lecture aims to demonstrate an understanding of the structure of biomolecules. Now, for our first presenter or discussant, she graduated cum laude of Bachelor of Secondary Education, major in Biological Science at Our Lady of Salvation College. At present, she is a teacher at Naga National High School of Department of Education, Albay. To discuss Introduction to Macromolecules and Carbohydrates, please welcome Ms. Aleth Rose and Canale.
Once again, hello everyone. This is Teacher Alat, and let's explore the wonders of biomolecules of life. Let's go. But first, let me present to you our objectives for this session. This 1 hour session should enable each participant to demonstrate an understanding of the structure of biomolecules and recognize the major categories of biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. Specifically, we are interested in finding out what they do for living things, what they generally looks like, what their monomers are, and how they may help the body gain energy to sustain life. Are you familiar with this pyramid? Actually, it's an inverted pyramid. Are you familiar with this one? Well, if you are familiar, this is the hierarchy of biological organization by Carl Linnaeus. Interpreting the pyramid and focusing on molecules, it emphasizes that a cell is made up of molecules and a molecule is made up of atoms. That's the simplest way of putting it, but which is complicated is that cells are made up of even complicated structures, which are the molecules. But first, let's define a molecule. What is a molecule? Well, molecules are the smallest particle of all substances that retains all the properties of the substance and is composed of one or more atoms. Knowing and understanding what molecules are, we are eager as well to understand what are the specific types of molecules do living things require. How are these molecules formed? And what functions do they serve? So now let's find out. Molecules are not only limited to living things, take note, but these also exist in non-living things. But our focus for now is to elaborate those different types of biomolecules, specifically those molecules that help sustain life. Like, what type of molecules are present in our food? Well, biomolecules is a short term for biological molecules, also referred as the molecules of life. So don't be confused. Biomolecules or biological molecules or molecules of life are the same. These are molecules that are involved in the maintenance and metabolic processes of living organisms. These non-living molecules are the actual foot soldiers of the battle of sustenance of life. Biomolecules are ranged from small molecules to large macromolecules and further classified into two. This includes the macromolecule and the micromolecules. What are the difference between the two? Well, referring only with the prefixes macro means large and micro means small, we can clearly simply define that macromolecules or polymer are large molecular molecules necessary for life that are built from smaller organic molecules known to be monomers or micromolecules. So therefore, macromolecules starts first with the micromolecules. There are also four major classes that falls under biological macromolecules, and this includes the carbohydrates, the proteins, the lipids, and the nucleic acids. Each is an important cell component and performs a wide array of functions. Combination of these macromolecules make up the majority of the cell's dry mass. So on the other side naman, we have the micromolecules as the monomers, or meaning they are the building blocks of each macromolecules respectively. Like, for example, the building blocks of carbohydrates are simple sugars, and so on and so forth. All Let me give you a short overview of biomolecules that speakers will later discuss to you. Again, what are the four major classes of macromolecules? We have carbohydrates, lipids, nucleic acids, and proteins. All of the mentioned macromolecules from the earlier slides are said to be organic. Why organic? Because all compounds that contains carbon are considered to be organic. So as you can see from this concept map, these four macromolecules contains carbon. So therefore, in short, all of them are considered to be organic. Earlier, we already distinguished what is a macromolecule and what is a macromolecule. But now, let me give you a brief definition of a macromolecule. What is a macromolecule? Well, it is a large organic compound. Again, if we say organic, it contains a carbon atom, and it also called a polymers because it's made up of smaller building blocks called monomers, which are the micromolecules. And it is also a very large molecule consisting of many structural units linked together by what we call the glycosidic linkage. Later on, you will further understand what is this glycosidic linkage that I'm saying. All biological macromolecules are made up of a small number of elements like carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Here, have you ever wondered if these macromolecules were formed from micromolecules, then what is the process or reaction it undergoes to successfully form a large size molecule? What do you think? Well, as stated in that slide, macromolecules is formed through the process of dehydration synthesis. What is this dehydration synthesis? It is a chemical reaction that combines single molecule into a complex one by removing water, commonly known as H2O. This synthesis reaction yields to glycosidic bonds that strengthen the connection between the two molecules. Given the example, combining the two monosaccharides yields to one disaccharide and a water molecule. So this reaction already undergo a dehydration synthesis. As you can see in the product part, we have water and the disaccharide. In reverse, how are these macromolecules broken down into simpler form? Well, macromolecules again undergo a reaction and which separated or digested this large molecule into a simpler form, and this is what we call the hydrolysis reaction. What is this hydrolysis reaction? This is a reaction wherein it adds water molecule to splits or to break the bond between the two bonds between to summarize, monomers or micromolecules are made into polymers by dehydration reactions, and in reverse, polymers are broken down into monomer by a hydrolysis reaction. Let's talk more about one of the major class of macromolecules, which is the carbohydrates. Did you know that carbohydrates is the most abundant macromolecule in nature? Yes or no? Yes, it's true. It is the most abundant macromolecules in nature because it is the most abundant dietary source of energy, but not just energy, but also a major structural component of all living organisms. And I'm sure everyone is familiar with carbohydrates known as carbs, especially if you have friends or relatives who's on a diet because they commonly mention about carbs. Correct me if I'm wrong. So now, we will now discuss further why carbohydrates are essential part of our diets. Biomolecules is consist mainly of carbon, hydrogen, with nitrogen, oxygen, sulfur, and phosphorus. But in carbohydrates, only these three elements exist: carbon, hydrogen, and oxygen. From the word carbohydrates, we can simply define carbohydrates as a hydrated carbon. As you can see from this illustration, carbohydrates contains one carbon atom, two hydrogen atoms, and one oxygen atom. Carbohydrates is represented by the above stoichiometric formula, where N is the number of carbon in the molecule. Therefore, the ratio of carbon to hydrogen to oxygen is 1:2:1 in every carbohydrate molecule. So, what do carbohydrates do for living things? What are their roles? Well, one of the major functions of carbohydrates to living organism is that carbohydrate serves as fuel and building material. They are the main source for the body to gain energy because they are our fuel. So when you completely cut carbohydrates out of your diet, you could put yourself at risk for nutrient deficiencies if you don't replace those nutrients with other food sources. And did you know that almost of the marathon runners eat carbs the night before the race? This is what they term as carb loading or carbohydrate loading. It is based on the knowledge that our bodies stores carbohydrates as glycogen in the muscles and liver, and this stored energy is then used to fuel long bouts of energy like running a half marathon. Another function of carbohydrate is that it provides a structural support in all living organisms. They make up the cell wall in plants, fungi, and even the exoskeleton of arthropods, and even crustaceans. Imagining without these structures like cell wall, without cell wall, all the functioning of all cell organelles present inside the cell would be affected as diffusion of various substances would not occur. So due to absence of turgor pressure, the cell will not bear the concentration of solution and will burst, as well as the absence of exoskeleton in arthropods, like crabs and shrimps. If they don't have exoskeleton, it's very vulnerable for them and prone to danger if they don't have that outer covering. So, see, the role of carbohydrates are amazing and yet meaningful to each of us. So, where are carbohydrates mostly found? Carbohydrates are sugars, starches, and fibers that are found in a wide array of both healthy and unhealthy foods, especially found in fruits, vegetable, milk products, popcorns, soft drinks, and even cookies. So it's really difficult to eliminate carbohydrates entirely from our diet. So we cannot conclude that carbohydrates are only for nutritious foods. But also we have food that contains carbon in an unhealthy manner. Now, let's begin with the monomer units of carbohydrates. These are the three classification actually of carbohydrates. Starting off with first with the simple sugars, or also known as monosaccharides, meaning this contains only one sugar, one unit sugar, And one of the common examples of this are fructose, glucose, and galactose. On the other hand, disaccharides are composed of two units of sugars bind together by a glycosylated linkage. Examples are sucrose, maltose, and lactose. Lastly, the polysaccharides from the prefix poly, meaning it is composed of many sugar units. Common examples of this are starch, cellulose, and glycogen. Monosaccharides as the building blocks of all carbohydrates serves as a major fuel for cells and as raw material for building molecules. Glucose as energy source for cells, and fructose found in fruits, and galactose sugar in milk. What did you notice from the structure of the three monosaccharides? The glucose, the galactose, and the fructose. From here, from this illustration, what did you notice? Are they the same? Are they the same? No. They are all, if you have noticed, their chemical formula are the same, but in terms of the arrangement of atoms, they are different. So meaning if there is a different arrangement of atoms, therefore there is a different in terms of their characteristics. So this is what we call isomers. Glucose, galactose, and fructose are isomers. So now let's proceed to the second classification of carbohydrates, which is the disaccharide. From the prefix di means two, therefore disaccharides are composed of two sugar units bonded by a glycosidic linkage. What is this glycosidic linkage? A glycosidic linkage or glycosidic bond are covalent chemical bonds that holds together a glycoside. And this bond was formed after a dehydration reaction. So what is a glycoside? A glycoside is simply a ring-shaped sugar molecule that is attached to another molecule. Here are examples of disaccharides. We have maltose, sucrose, and lactose. Maltose or malt sugar is a combination of two glucose molecule which is common among wines. Another is sucrose or known as table sugar, which is a combination of glucose and fructose. And lastly, lactose or milk sugar which is commonly found on milk products, is a combination of glucose and galactose. So now, the third classification of carbohydrates, the polysaccharides, represented in a chemical formula or in an empirical formula, C6H10O5N, where N is the number of the carbon molecule. What is a polysaccharide? Polysaccharides, from the prefix poly again, it means many, so therefore polysaccharides are composed of many sugar units, and these are the polymers of sugars which serves as storage and structural component of every living organism. The structure and function of a polysaccharides are determined by its sugar monomers and the positions of glycosidic linkages. As you can see, this is an example of the structure of a polysaccharide. And we can compare it, or if you have noticed, polysaccharides are similar to a necklace, wherein each bead or each carbohydrate molecule is the bead, and the necklace as a whole will represent the polysaccharide, which is bonded by a glycosidic bond. Starch and glycogen falls under polysaccharides. They are considered as one of the most abundant carbohydrates in nature. Why? Because starch are found in plants and glycogen for humans and animals. Starch is stored in a cell organelles called amyloplasts. The leaves of a plant make sugar during the process of photosynthesis. So when plants are making sugar on a sunny day, they store some of it as a starch. When the simple sugar needs to be retrieved for use, the starch is broken down into smaller components, which are the glucose. On the other hand, glycogen is stored primarily in the cells of the liver and skeletal muscles. When the body doesn't need to use the glucose for energy, they are stored in a form of glycogen. So in short, starch and glycogen are similar, but in a way na starch is the stored energy for plants and glycogen is the stored energy for animals and humans. This cellulose, chitin, and peptidoglycan are examples of polysaccharides but considered to be structural molecules. Why structural molecules? Well, let's find out. Cellulose is a type of a polysaccharide that commonly found in plant cell walls, and it was said that most organisms wasn't able to break cellulose bond, but there are specific enzymes that can help to break down this bond, but not all organisms can able to break this bond. Another structural molecule that falls under polysaccharide is chitin. What is this chitin? It is a structural molecule that plays a vital role among arthropods and some crustaceans. What are these? What are the chitins? Well, they are the outer shell or their exoskeleton. It was said most of the success of arthropods is because of their exoskeletons. So imagine what will happen to them if they don't have exoskeletons. For example, think of a crab or shrimp. What do you think will happen? It's very dangerous for them. Why? Because it's one of their means of protecting themselves from predation. It also functions as a protective covering of their internal organs and muscles from the external environment. And at the same time, it helps them to achieve efficient locomotion. So, imagine if there would be no chitin, if there would be no carbohydrate containing this kind of structure. So, it's very difficult for them. Diba? And lastly, the one of the structural molecule that falls under polysaccharide is the peptidoglycan. Peptidoglycan is the main component of the cell wall in most bacteria. Because bacteria are unicellular, peptidoglycan gives strength to the outer structure of the organism. It is also involved in binary fission rather, which is how bacteria reproduce. So, who can imagine how the scope of carbohydrates? It's so broad, yet amazing. So, it really exists, or we can therefore conclude that carbohydrates really exist everywhere. It's abundant. It really is. It really is. I'm sure with that. So let me end this part with a short message taken from the fundamental of human nutrition. So think first about the nutritional content of food in your plate, because awareness of the contents of your consumption is the first step in moving towards a healthier diet and therefore a healthier body and a healthy living. So this concludes my topic about carbohydrates. Once again, good morning and God bless.
Our second discussant also graduated cum laude of her batch from Bachelor of Secondary Education, major in Biological Science at Bicol University, Tabaco Campus. She is now a full-time Department of Science and Technology, Capacity Building Program for Science and Mathematics Education scholar at Bicol University. To discuss lipids and proteins, please welcome Miss Christine Joy B. Vista.
Good day everyone. So teacher Alat is done discussing about the first group of biomolecules, which are the carbohydrates. So this time I will be discussing about the second and third group of biomolecules, which are the lipids and the proteins. So upon discussing, we will be able to answer the following questions. What are lipids? What are the roles of lipids? What are the different types of lipids? What are the structures of lipids? And what are the importance of lipids? So let us start getting to know about lipids. So primarily, it has the job of storing energy for later use. So they can be found in hormones and cell membrane components. So here's an illustration of a cell membrane structure. So one of the most important picture of this is the lipid bilayer, or commonly called phospholipid bilayer. So the main role of lipids in cellular functions is in the formation of the permeability barrier of cells and subcellular organelles in the form of a lipid bilayer. So lipids have different structural types such as carboxylic acids or the fatty acids, triglycerides or neutral fats, steroids, and waxes to name a few. And naturally occurring esters are lipids that contain one or more long-chain carboxylic acids called fatty acids. So these are insoluble in water but soluble in non-polar solvents. So we have here two types of fatty acid, the unsaturated and saturated fatty acid. So unsaturated fatty acid are those long-chain carboxylic acid that has double bonds, while saturated fatty acids are those with single bonds. So let us try to think of it. For example, Alex accidentally mixed oil and water. Then he observed that they do not mix. So why do you think so? So the oil and the water do not mix because they do not have the same polarity. Also, oils are composed primarily of long hydrocarbon chains, and they are formed reaction between an alcohol and one or more long-chain carboxylic acids. And oil and water is said to be immiscible. So water is a polar molecule. And its structure means that it has a positive charge on one end and a negative charge the other end. So water molecules stick together because the positive end of the water molecule is attracted to the negative end of another. Then the structure of an oil molecule is said to be nonpolar. So its charge is evenly balanced rather than having one positive and negative end. So this means that oil molecules are more attracted to the other oil molecules. The same with the water molecules. Its molecules are attracted to each other than with the oil molecules. So the two will never mix. So the most abundant of the lipids are the fats and oils, also called triglycerides. So these are the excellent long-term energy storage molecules because they will not mix with water and break down. It is made of a glycerol backbone. When we say glycerol, it is a simple sugar alcohol. Also, the triglyceride structure is consists of a three fatty acid chains attached to it. So the presence of double bonds in the fatty acids lowers its melting point. Just like in the examples here in the list. So here are some of the common fatty acids. There's the lauric, stearic, palmitic, stearic, palmitoleic, oleic, linoleic, and linolenic acid. So we can see that some of these fatty acids have double bonds, and it has something to do with its melting point. So the presence of double bonds in the fatty acids lowers its melting point. That's why at room temperature, lauric acid is solid while linoleic acid is liquid. So let us discuss about fats. So fats are solids at room temperature and contain saturated fatty acids. Saturated hydrocarbons contain single bonds and they are produced only by the animals. For examples are the animal fats such as lard and butter. So these are derived from fat tissue of the animals. While oils are liquids at room temperature and contain unsaturated fatty acids, unsaturated hydrocarbons contain one or more double bonds. So for examples are the oils produced by plants such as vegetable oil, corn oil, and olive oil. So sometimes we prefer to buy a product in solid form rather than in liquid. So which do you prefer? Spreading margarine on a pedestal or pouring oil on it? So margarine is more acceptable to consumers when it is solid because it looks more like butter. However, margarine is made from vegetable oils that are liquid at room temperature. The oil can be processed to form solid margarine. So how is this done? So it is done through a process of hydrogenation. When we say hydrogenation, it is a process in which a liquid unsaturated oil is turned into solid fat by adding hydrogen. Hydrogenation converts vegetable oil into solid or semi-solid fats, such as those present in margarine. Then changing the degree of saturation of the fat changes some important physical properties, such as the melting range, which is why liquid oils become semi-solid. But which is more preferable? Which is better, oil or solid fat? The solid fats contain more saturated fats or trans fat than oils. Saturated fats tends to raise low-density lipoproteins cholesterol level in the blood, which in turn increases the risk of heart disease. So can we consider fats good or bad? So it depends. If you eat in moderation, fats are good sources of body fuel. They are considered good emergency food and are efficient storage system. So let us avoid eating too much foods with trans fat. It is a byproduct of hydrogenation, which turns healthy oils to solid to prevent them from becoming rancid. So trans fat have no known health benefits in that there is no safe level of consumption. So let us avoid taking too much of the bad fats, such as those coming from fast food chains, such as burgers, french fries, pastries, etc. And let us take more of the good fats, the olive oil, corn oil, avocados, etc. However, an excess quantity of fats is not good for the heart. So why? Why it is? So the reason why too much fats are not good for the heart is because they tend to clog arteries and overwork the heart. So the veins get smaller and smaller with all the fatty part of the food, and the blood can't pass, so you get a heart attack. While carbohydrates are the main source of energy in your body, your system turns it to fat as a backup energy source when carbohydrates are not available. So vitamins A, D, E, and K cannot function without adequate daily fat intakes since they are fat-soluble vitamins. When we say fat-soluble vitamins, those are the vitamins that dissolves in fat and are stored in tissue, so the body has access to them as needed. But if you don't meet your daily fat intake or follow a low-fat diet, absorption of these vitamins may be limited, resulting in impaired functioning. So steroids are another class of lipids whose molecules are composed of few strings of atoms. So the most important steroid is the cholesterol. It is a steroid because of the presence of alcohol or the hydroxyl functional group. It is found mainly in animal cells, although cell membranes of plants may contain small quantities of cholesterol, as well as its major derivatives, sitosterol. So this is a unique structure of cholesterol which consists of four linked hydrocarbon rings forming the bulky steroid structure. So there is a hydrocarbon tail linked to one end of the steroid and a hydroxyl group linked to the other end. So cholesterol is known as a sterol because it contains an alcohol functional group OH. Then the cholesterol is present in most animal membranes with varying amounts but absent in some prokaryotes. So cholesterol plays an important role in eukaryotes and is especially abundant in cell membranes of animal cells. Small amount of cholesterol can also be found in the membrane of some organelles inside the cells, such as the mitochondrion and the endoplasmic reticulum. So it is not only abundant in cell membrane but also in brain tissues of the nervous system. So an important nerve cell, myelin, covers nerve axons to help conduct the electrical impulses that make movement, sensation, thinking, learning, and remembering possible. So it is said cholesterol is the best brain food. So studies have shown that cholesterol was found to be the most important factor in the formation of synapses which greatly affect our memory and learning ability. So animals are able to use cholesterol to synthesize other steroids like cortisol, testosterone, and estrogen. However, excessive levels of cholesterol in the blood can lead to deposits in the arteries of the heart. These arterial deposits are a leading cause of a heart disease. So we are done discussing about lipids. So let us proceed to another biomolecules, which are the proteins. So we will be answering the following questions. What are proteins? What are the roles of proteins? What are the different types of proteins? What are the structures of proteins? And what are the importance of proteins? So proteins are made up of elements carbon, hydrogen, oxygen, nitrogen, and sulfur. Egg white, fish, meat, and cheese are foods rich in proteins. So proteins are found in all living cells. So they are the most common molecules found in the human body after water and make up about 10% to 20% mass of a cell. So let's say you ate a protein-rich food. What do you think will happen to the proteins? So your digestive system will break the long protein chains into simpler substances called amino acids. So amino acids are called to be the building blocks of protein. So of the 20 amino acids in human protein, only 11 can be synthesized by the body, and 9 have to be supplied by the food we eat. So these 9 amino acids are also called the essential amino acids. So the adults only need to obtain 8 of them: valine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan. Then the ninth amino acid, which is the histidine, is only essential for infants. So your body doesn't store amino acids, so it needs a regular daily supply of these essential building blocks. So are non-essential amino acids still essential, or are they still important? So non-essential is a slightly misleading label because these amino acids actually fill essential roles. But since they are synthesized by your body, they are not essential part of your diet. Alanine, asparagine, aspartate, glutamate, and serine are non-essential. Of the 11 non-essential amino acids, 8 are called to be conditional amino acids. So when you are sick or under significant stress, your body may not be able to produce enough of these amino acids to meet your needs. So the list of conditional amino acids include arginine, glutamine, tyrosine, cysteine, glycine, and diproline. So here's a summary list of the three types of amino acids. Under essential amino acids, we have histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. While in conditional non-essential, we have the arginine, cysteine, glutamine, glycine, proline, and the tyrosine. While on the non-essential, we have alanine, asparagine, aspartate, glutamate, and the serine. So whenever you eat protein foods, you are supplied with amino acids for the rebuilding of your body system. So our body is composed of approximately 16% of proteins. So here's the amino acid structure. Amino acids are organic molecules that contain two functional groups, a basic NH2 amino group and an acidic COOH carboxylic acid group. So when two amino acids react with each other in an acid-base reaction, a peptide is formed. So the basic amino group of one amino acid reacts with the acidic carboxylic group of another amino acid, forming the peptide, and a molecule of water is lost. So the reaction shown in classified as condensation reaction because the amino acid molecules join together and then water is formed. So the bond formed is called peptide bond and the product is dipeptide because it is made up of two amino acid units. So longer chains are called polypeptides and chains of 50 or more amino acids are called proteins. So proteins are characterized by their primary, secondary, tertiary, and quaternary structure. So the kind of amino acids which make up the chain, the sequence in which the amino acids are arranged and the length of the chain distinguishes the primary structure of proteins. So primary structure is simply the linear sequence of amino acids in a peptide bond or protein. So the secondary structures of proteins refer to the coiling of the protein chain into a alpha helix structure, formation of beta sheets, or twisting into random structures. So these structures are the result of interactions between R groups, hydrogen bonding, or formation of SS bonds between chains. So protein molecules are so long that they automatically coil, fold, or twist. So the resulting shape is unique for each polypeptide in a particular medium at a particular pH. Also, an alpha helix and beta pleated shape form because of hydrogen bonding between carbonyl and amino groups in the dipeptide backbone. Certain amino acids have a propensity to form an alpha helix, while others have a propensity to form beta pleated sheet. Kita din na naman yan ni Masay.
Kada ano kaya din pag-save mo?
Control S, Control S.
So check mo. Pero mag-text ka dyan, balikan mo kung mag-gawa.
AX ko, girl. So here's a picture showing the primary structure and a secondary structure of protein. Then the tertiary structure describes the shape of the coiled chain when it is folded or hydrated in its natural state, or once all the secondary structure elements have folded together among each other. Interactions between polar, nonpolar, acidic, and basic R group within the polypeptide chain create the complex three-dimensional tertiary structure. So the last structure is the quaternary protein structure. It involves the clustering of several individual peptides into a final specific shape. So a variety of bonding interactions, including hydrogen bonding, salt bridges, and disulfide bonds, hold the various chains into a particular geometry. So quaternary structure consisting of more than one amino acid chain. So we have here another type of protein, which is the enzyme. So it is known as biological catalyst. These molecules speed up biochemical reactions without themselves being used up in the process. They are also highly specific. That is, they act only on certain molecules called the substrates or the reactants, while leaving the rest of the system unaffected. So the role of an enzyme can be compared to lock and key. So you cannot open a lock unless you find the right key. So the enzyme works for a specific substrate. So an enzyme attracts substrate to its active site. So catalyzes the chemical reaction by which products are formed and then allows the products to disassociate. So the combination formed by an enzyme and its substrate is called an enzyme-substrate complex. So we should realize how important enzymes are. So you must appreciate the role of enzymes in the body. Without them, chemical reactions in the body may be too slow to occur at normal condition and may affect the normal functioning of the different systems of the body. So learning all these things, we should realize how important biomolecules are, especially for the survival of living cells. Proteins are the primary building materials of your body. Your hair, skin, muscles, and enzymes are composed of mostly proteins. Then lipids are the primary long-term energy storage molecules of the body. So that ends my presentation. This is Teacher T. Thank you so much for listening. God bless everyone.
Lastly, our third presenter. She graduated cum laude from Bachelor of Secondary Education, major in Biological Science at Bicol University, Tabaco Campus. She is a former teacher at Caroline A. Institute of Technology Incorporated, and now also a full-time Department of Science and Technology Capacity Building Program for Science and Mathematics Education Scholar at Bicol University. To discuss properties of nucleic acids, please welcome Ms. Jessica Jane Bobbies.
Hi guys, this is Teacher Jess, and join me as I unlock your queries on the last type of biomolecules, the nucleic acid. So a living organism is composed of biomolecules. A biomolecule makes up an organism, and we all are examples of it. Imagine you are full of biomolecules. It is also composed of building blocks called monomers. These monomers are interconnected to each other to form the biomolecules. So there are four biomolecules. We have carbohydrates, lipids, proteins, and our topic, which is the nucleic acid. They are very essential to sustain life and its processes. So from your previous lessons, you have discussed the structures of biomolecules. So in carbohydrates, we have carbon, hydrogen, and oxygen. Same as your lipids, you have carbon, hydrogen, and oxygen. So for proteins, we have carbon, hydrogen, oxygen, and nitrogen. And in nucleic acid, we have carbon, hydrogen, oxygen, nitrogen, and phosphorus. So it might be very confusing. So let me leave you a few hints to remember them easily. The CHO, CHO, CHON, and CHON. So carbon has CHO, lipid has CHO, protein has CHON, and nucleic acid has CHON. So it is easier to understand and remember. So here are some food rich in carbohydrates. So here are some foods rich in lipids. And here are 4 proteins. So nucleic acids are polymers, which means it has many chains of monomers. The obvious example of nucleic acid is DNA and RNA. So DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. The major functions of nucleic acids are it holds genetic information, the reason why you look like your parents, and it codes for proteins. So the question is, what are the foods rich in nucleic acid? So here are the foods rich in nucleic acid. What can you observe? Correct. They are either animals or plants or a product of them. Plants and animals are living organisms. So let us make a few analyses why they are rich in nucleic acid. First, DNA and RNA are examples of nucleic acid. And all living organism has DNA. Therefore, anything that is once living is rich in nucleic acid. Makes sense, right? So let us now move on to the structure of nucleic acid. The monomer or building blocks of nucleic acid is nucleotide. A nucleotide is composed of sugar, phosphate group, and nitrogenous bases. So when these three combines, it forms a nucleotide. So here is an illustration of a nucleotide that has parts that are connected. So we have the phosphate group, sugar, and the nitrogen base. So let us now discuss sugar. A nucleotide is composed of a 5-carbon sugar or a pentose sugar. Since it has 5 carbons, it is called pentose, coming from penta, which means 5. The 2-pentose sugar in our body is called ribose and deoxyribose sugar. So what is the difference between the two? Correct. There is no oxygen in C2 in deoxyribose. It is because it has a prefix deoxy, which means the removal of oxygen. Proceeding to phosphate group. So oxygen binds with phosphorus through an ester bond. So it is composed of phosphorus and 4 oxygen. When a phosphate is added to a nucleoside, the molecule is called a nucleotide. Nitrogenous bases. Molecules that contains nitrogen and act as a base. Nitrogen binds with C, H, and O, or carbon, hydrogen, and oxygen to form ring-like structures. So here are the 2 ring-like structures. We have pyrimidines, which is the 1 ring, and purines, which is the 2 rings. So under pyrimidines, we have cytosine, thymine, and uracil. And for purines, we have guanine and adenine. The photo shown is an example of many nucleotides connected. The sugar phosphate backbone is composed of sugar and phosphate bonding to each other to continue the chain. Shown are the nitrogen bases on the right side of the illustration. They bind with the complementary pairs, which are Cg and At, through hydrogen bonds. C stands for cytosine, G is for guanine, A is for adenine, and T is for thymine. Now the sugar phosphate backbone on the left and right seems to be non-parallel. The sugar phosphate backbone on the right side are chains of inverted sugar phosphate. So the given example a while ago is a DNA. So here is now the difference between DNA and RNA. DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. DNA has double-stranded sugar phosphate, while RNA is usually single-stranded. And DNA has deoxyribose sugar, and RNA has ribose sugar. And for the nucleic base, DNA has cytosine, guanine, and adenine, and thymine, while RNA has cytosine, guanine, adenine, and uracil instead of thymine. So let me give you some questions. Can you imagine if there was no DNA that holds genetic information? What do you think are the possibilities? Do you think you'd look differently from your parents? Or will all of us look exactly the same? What do you think? Why are these DNA and RNA important? 1. DNA holds genetic information. 2. RNA transmits and expresses hereditary information. And 3. It codes for. So here are the key concepts you need to remember after the 4 topics. So first is macromolecules are polymers built from monomers. Carbohydrates serve as fuel and building materials. Lipids are a diverse group of hydrophobic molecules. Proteins include a diversity of structures resulting in a wide range of functions. And nucleic acids store, transmit, and help express hereditary information. Here is the concept map for you to remember the 4 structures. Carbon compounds includes carbohydrates, lipids, and nucleic acids, which consists, for carbohydrates, you have sugar and starches, lipids has fats and oils, nucleic acids has nucleotides, and protein has amino acids. And this contains carbohydrates has carbon, hydrogen, oxygen, lipids has carbon, hydrogen, oxygen, nucleic acids has carbon, hydrogen, oxygen, nitrogen, and phosphorus, and proteins has carbon, hydrogen, oxygen, and nitrogen. So that ends our presentation. I hope that you have learned something from our 1-hour session. Let us always remember to be mindful of our health. This is Teacher Jess, signing off.
Thank you, participants, learners, for participating in this event. We hope that you gain a volume of knowledge in terms of biomolecules, specifically carbohydrates, lipids, proteins, and nucleic acids. We hope that you stay safe and you stay healthy at this time.